Hello students, welcome to the biology class. Today we will study the topic from your chapter, that is reproduction. Chapter name is sexual reproduction in flowering plant, and the topic is development of female gametophyte. In this topic we basically do how the development of female gametophyte will take place, which is also commonly known as embryo sac. In order to do the process, that is the development of female gametophyte, we firstly make the diagram of an ovule. We will study from the strategy means reproduction fund and is development of female gametophyte. This topic is you how the development of female gametophyte will stay, which is also known as embryo sac. In order to do this development of female gametophyte, we firstly make the diagram of the ovule. And already this form as symbol. These are the units, and this is the gland, and this is the microgland of an ovule. We have to say on this section is called as cellular mass. But I think the development female gametophyte, that is the part, there are number of cells, and of these cells, one cell and it is called as the microglia cell. Now we take this outside structure and we see how the development of this bigger cell will take place. Whenever we do development of female gametophyte, we have to realize that we have definite cells, the most and the lower portion is micro. Now we have the microglia cell here, that we get my biggest cell. Now see what I will say is this cell. Now this cell will go. As you know, that all these values are the ones that will transmit into the cell. Those cells are done. We have this here. And this is out of 3 or 4 and look at this cell from the out of these 4 makes these cells which are not and we have 1 cell here. So for now we take a of the ovule, we will take the cell, which we get the process all the white out of these cells, 3 makes cells and only the white that is towards present as a high. Now see what are these other reasons here was and now about this. This makes 4 cell and go microglia. So you have this that is initially now this yes and go. And as we went, microglia is comprised of 2 phases. First is called as catalysis, which nucleus device, and second process is cytokinesis, in which the cell now this figure 4 will undergo 3 catalysis. 3 catalysis means nucleus of this figure 4 divide 3 and the how this reason is figure 4. The first mitotic nucleus catalysis is formation of nuclei. Both are haploid. In second karyokinesis, second mitotic division, second karyokinesis, this cell will nucleus is divided again and there is formation of 4 nuclei. Now this cell again undergoes third mitotic division and that is third karyokinesis, and we will get eight nuclei. So now we have a megaspore cell which undergoes 3 karyokinesis, that is the mitotic divisions, and ultimately we get a cell which has 8 nucleuses. Now we will see that what are the changes that takes place in this structure. So I have to rub it. Now this cell has 8 nuclei. Now these 8 nuclei will rearrange themselves towards the clavell and or the microglia. Now we will see how their rearrangement of these nuclei takes place. Okay, so in next outer microbial end. And two nuclei will remain in the center of that cell. Now, the next event, there takes place, which means that there is formation of the cell wall around these nuclei. Now, the structure changes that occurs in this cell are, we will keep these two nuclei inside in the center. We will make membrane around these cells at the basal end and the microbial end. Now, if you look carefully in this diagram, I am going to name them. What are the cells that are formed on the basal end are called, or what are the cells that form on the microbial end are called. The cells that are formed, three cells that are formed on the basal end are called as antipolar cells, and the cells which are formed on the microbial end, these two cells are called as synovids, and the center cell is called as egg. And collectively, this structure, that is two synovids and a center egg, is called as egg apparatus. And these all cells are haploid, and their center, and in the center there are two nuclei present. These are called as polar nuclei. If you see carefully, you will see that the name of this structure is now called as embryo sac, because there are presence of 1, 2, 3, 4, 5, 6 cells and one central cell. So this structure will become how many cells and how many nucleated? This structure will become 7 cell, 8 nucleated structure, and this structure is called as embryo sac that we are talking about, that is the female. The structure that formed is 7 cell and 8 nucleated. Let us count the cell. 1 cell, 2 cell, 3 cell, 4 cell, 5 cell, 6 cell, and the center cell that is having the 2 polar nuclei. And now we will count the nucleuses. 1 nucleus, 2, 3, 4, 5, 6, 7, 8. Now we have the condition 7 cell, 8 nucleated condition in embryo sac. In many of the angiosperms, we can say that in the maximum of the angiosperms, that is the form, the embryo sac is in 7 cell, 8 nucleated condition. It is also called as polygonal embryo sac, monosporic embryo sac, are female gametophyte, are 7 cell, 8 nucleated embryo sac. So you have to remind all these things, that how the development of the angiosperms will, how the development of female gametophyte will take place in angiosperms, where we have started from a single megaspore mother cell which undergoes mitotic division. We get the 4 megaspore mother cells, out of which 3 are degenerate, only 1 remains, which undergoes again mitotic divisions, that are 3 karyokineses, and ultimately we get the 8 nucleated 7 cell condition. So this is for our, which comprises, you have completed your topic, that is development of the female gametophyte, that is embryo sac. Thank you.